Okay. Hello, everybody. Welcome to the Let's Get Lit series. This is number 3, Bacteria versus Cancer. And my name is Alan, and I'll be leading this one with my friends here today. So,
Colin and Angel.
All right, let's get started. Today is called Bacteria versus Cancer, which is something I feel like people don't usually put together, right?
It's like a blockbuster.
A blockbuster film.
Yeah.
Bacteria versus Cancer. Yeah. But hopefully this will give viewers an appreciation of the applications of bioengineering. This is a very clearly medical application. So to get started, this is the paper we will be going over. It's called Programmable Bacteria Induce Durable Tumor Regression and Systemic Anti-Tumor Immunity. A really big mouthful, like most papers are, but we'll go through it. First of all, Angel, what is the cancer cell?
It's basically a cell that constantly reproduces and kills itself, which is very negative against other cells.
Yeah, it definitely reproduces a lot.
Yeah.
And in fact, it's so uncontrollable that it forms this tumor mass,
Mm-hmm.
right? And it might kill the host.
Yeah.
And in fact, what you see in this diagram here is indeed a tumor cell begins to replicate by mitosis. It divides and divides and divides. And what's interesting is that in the middle, there tends to be a lack of oxygen. It's hypoxic in the middle. And that makes sense because if you think about oxygen, it diffuses into certain spaces, which is how cells acquire that oxygen. But if you have a lot of cells growing really, really fast without the necessary blood vessels, the middle becomes hypoxic. And so what cancer cells sometimes do is they'll form blood vessels. And that process is called angiogenesis. So they literally force the host's body blood vessels to grow into the tumor, which is
Kind of crazy.
kind of gross. Yeah, kind of crazy. But what's interesting is that people can take advantage of this hypoxic environment. And so I say this because some bacteria actually localize to hypoxic environments. So is it that should be like a foreshadowing effect right there, right? So bacteria localize to hypoxic environments, hypoxic meaning low oxygen. So another sort of side tangent on cancer cells. Cancer cells can evade the immune system. Essentially, there's this protein that certain cancer cells overexpress, and that protein is called CD47. It's a membrane protein. And the membrane protein essentially binds to another protein on various immune cells called SIRP-alpha. And that binding, it says, don't eat me. Don't eat me.
Don't eat me.
Right, yeah.
It's a nice camouflage.
I'm watching Attack on Titan. So it's like, don't eat me, don't eat me. If I was a cancer cell, I'd be like, don't eat me. Obviously no one wants to be eaten, right?
Mm-hmm.
But of course, normal cells, when they malfunction, they undergo apoptosis, of course. And certain immune cells then come around and eat up their debris. But in this case, the cancer cells, they sabotage that machinery, and they essentially say, don't eat me, so they survive and they grow into a tumor. Colin, knowing this, how might we design a therapeutic to kill tumor cells?
There's a couple ways you can think of.
Mm-hmm.
One way would to be like a way you could block the CD47.
Yeah.
You're a genius, Colin.
My degree's paid off.
You're a genius. Exactly. You block the interaction, which makes sense, right? If this binding says don't eat me, if you block that binding, then you will undergo phagocytosis. The immune cell will eat the cancer cell if you use something to sequester or bind to the CD47. So these are really useful as potential therapeutics. So this is now an eat me signal, because now the immune cell sees the cancer cell and wants to eat it. So the authors use that to practice. They don't use a full antibody. They use part of an antibody. Specifically, we call that a nanobody, and it's the very tippity-tip portion of the antibody, the part that binds to the CD47. So now that is the tangent on cancer. Now I'll go on another tangent on bacteria. You'll see how these two combine in just a bit. Bacteria we oftentimes see them as individual organisms, I feel like. You know, we talk about how humans have a society. There's no bacteria society. Or is there? Or is there, right? There is. I mean, bacteria form communities all the time. They form biofilms, for example, and they aggregate together. Sometimes bacteria only produce certain virulence factors that cause a lot of inflammation when they're in high enough concentration. So they sense each other, and that sensing is called quorum sensing. And they use that, they use these molecules called autoinducers to kind of sense each other, if that makes sense. So, I mean, we're communicating right now by verbal, right, by words. So we're using sound waves to communicate. Now, autoinducers are a chemical form of communication between bacteria. They can kind of think of it as chemical talking to each other. So when there's low autoinducer concentrations, the bacteria does individual behavior because they think they're alone. But if there's a lot of autoinducers, the bacteria now knows, oh, I have some friends around me. I'm going to now do a group behavior, like forming biofilms or, you know, spreading virulence factors, etc. And by the way, this AHL, acyl homoserine lactone, is what it stands for. That is the autoinducer. It's a molecule that allows cells to communicate to each other. And it's produced by this thing called LuxI. LuxI is an enzyme that can produce this talking chemical. And now notice how we can engineer this. This is a bioengineering channel, after all. We can engineer this system to make what we want only in the presence of high concentrations of AHL. In other words, this circuit, this target pathway, will only activate when there's enough bacteria present at a certain point. And here's how it works. The AHL molecule, which by the way, I'm a chemistry major, so I have to show the structure. This is AHL, right? With various. AHL is a molecule with various R groups, so there's a group of AHL, a group of autoinducers. And the AHL molecule can essentially go in and out of the cell. It diffuses around. No, it's very free. And it also binds to this purple box. We'll call it LuxR. The LuxR protein, once it's bound to AHL, can then start to transcribe other genes. So we can then engineer this green gene to be whatever we want. For example, an anti-tumor protein. No. So this is combining together quorum sensing and cancer therapeutics into one project, which is really cool. By the way, just a fun fact, bacteria are often used in bioluminescence. So for example, that bacteria will only sort of glow in the dark if you have enough of them together. So a lot of ocean creatures, they glow in the dark because of bacteria. And they do that by quorum sensing. They sense sort of where the highest concentration of bacteria are, and then they can start, bioluminescing, which is pretty cool.
Party.
Yeah, exactly. Part of a bacteria party.
Bacteria party.
Exactly. They have their whole, frat LED lights and everything. Okay, let's get into the paper's design. They know about the CD47, this don't eat me signal. They know about quorum sensing. The beauty that they sort of see in that is you can put those together into one system. Here's what they do. They engineered this SLC. It's called a synchronized lysis circuit. What that means is you have this gene here that makes AHL. As we said, the AHL can go in and out of the cell as it wishes. The AHL can then cause expression of more AHL. Of more enzyme that makes AHL. And so that is what we call a negative or positive feedback loop. Positive? It is positive, right? That's a positive feedback loop. And so, AHL binds to this protein, which makes more AHL. That's a positive feedback loop. They also engineer a negative component to that too. And obviously that's just the second part of this. There is this protein called protein E from this bacteriophage that causes bacteria to lyse. Lysis means just essentially exploding. So this is the protein that causes bacteria to explode. And if a bacteria explodes, Can it then make AHL?
No.
No, just a negative feedback loop. Now, the beauty of this is they actually optimize this such that it only lyses after a certain threshold of protein is made. In other words, once a quorum is reached. So what we see is in these cells that have this synchronized lysis circuit, the cells will grow, exponentially growing. But then once they reach a certain point, once enough of this AHL is around and the bacteria sense, oh, I'm around other friends, I have a quorum, this threshold is reached and all the bacteria just die. Like this. And they drop. And they die. And they grow more and they die again. And that cycle repeats over and over again. And that's not seen in the bacteria that don't have that circuit. Now, I haven't talked about the anti-cancer part yet, though. So they don't show this in the diagram, but they have another plasmid inside that codes for this CD47 nanobody, which you'll recall is a protein that binds to that don't eat me signal that essentially erases the don't eat me signal. So you can imagine the bacteria, after being injected into the tumor, will accumulate in the hypoxic tumor environment, will grow sufficiently to reach the threshold, and they'll all explode, and they'll shoot out their guts, and their guts have this anti-tumor protein. And then that anti-tumor protein will then bind to the tumor cell receptors, and then the body's immune system can then do the rest. That's a pretty neat idea.
Yeah.
Right? So any questions so far about this system?
I was wondering, it seems very inaccurate. Like it seems like a mitigation rather than like a full accurate, which is what most of these cancer, like that's the biggest problem, is accurately getting rid of all of these cancer cells. So I just want to know like how accurate, or what's the percentage that these cancer cells like die off whenever this happens?
Yeah, good question. I got a bunch of graphs.
Okay.
Showing you just that. Right now I'm just storytelling.
Okay.
I'm just telling the story. But then your question is, where's the data?
Yeah.
Like a true scientist, right?
Yeah.
Where's the number? So plug into that right now.
Okay.
Yeah. Not now actually. This is just a proof of concept, essentially principle.
Okay.
So what this is showing is the bacteria guts. That's what this is. As you increase the number of bacteria guts, it's just showing that the CD47 is being bound to. Because this dye, which competes for that receptor, is unable to compete with the nanobody produced by the bacteria. And by the way, these cells that are being used are called A20, and they're B-cell lymphoma. They're a cancer cell line. And also what's interesting is that when you have bacteria with this synchronized lysis circuit, and bacteria with the synchronized lysis circuit, and also the nanobody, you increase the degree of phagocytosis of these A20 cells, which makes sense because this is getting rid of the don't eat me signal. This is interesting though, Colin. I mean, if you look at here, why would there be an increase in phagocytosis if there's no nanobody, just the lysis circuit itself?
Yeah, that is interesting.
That's interesting. And it's pretty significant too, right, these stars?
Yeah. This was a question I had earlier maybe. It's like, if a bacteria is exploding its guts, wouldn't you get an immune response just based off of that as well, right? And do you think that's what's happening there?
Exactly.
Yeah.
Exactly. These bacteria are specifically E. coli that are non-pathogenic. And so you're right that might be a concern, but they chose a strain very carefully with that regard. But you're right, there is still this response. And I think what the word that you're trying to get at, I think you pretty much described it actually, is called adjuvant. Have you heard of that
I have not.
word before? It's used in have you heard of adjuvants before?
I have not.
It's a word used in vaccines. In vaccine science, you have the antigen itself that produces sort of the cells that are specifically targeting against that antigen, and you also have the adjuvant. The adjuvant is kind of like the thing that enhances the immune response.
I see.
So the bacteria, as you said, they're foreign. They actually enhance the immune response to increase phagocytosis against the cancer cell.
Right.
So it's actually like a helpful thing.
Yeah.
It turns out. So it's pretty interesting. But you're right, there might be some other effects that we don't really know about. But this is interesting just as an adjuvant effect. back to your question. Does it work? It works. Here is a mouse with 2 tumors, with 2 cancers on both flanks. You inject these bacteria into the tumor, and you see that only the cells, only the mice that were injected with the bacteria that contained the synchronized lysis circuit and the nanobody died. Right? The cancer was there, and it went down. It died.
Yeah. Extremely effective.
Right. So this is your data right here.
Yeah.
So it works in these mice models with this A20 cell model cell line, the cancer cell model.
What happens if you start like at a higher tumor volume?
That's a good question. Because I mean, your question is suggesting sort of if we have a patient with sort of late stage, right? I don't think they show that. That's a good point. Yeah. So
another question, maybe you'll get to this as well, is like the ability for it to localize. Like, do I have to inject it right into the tumor?
I will get to that. I will get to that. Yeah, exactly. So these are IT, intratumoral injections, and it seems effective, at least with this initial tumor volume, right? The number of metastases also decreases. So metastases, are you familiar with that term?
No.
This is when a tumor spreads. So tumors can spread, and that's oftentimes one of the very deadly things about them. They can get into your blood vessels, and they can localize elsewhere. So for example, if you have lung cancer, that might localize and spread to your liver or your brain, and that might not be good. And so you notice that the number of metastases also decreases, probably because the tumor itself died, right? Which makes sense. Furthermore, these mice also survived for longer. that's important, right? That's important. And so these mice treated by injecting the bacteria into the tumor survived by a significantly longer time. And if you re-challenge them, meaning they had the cancer died, the tumor volume decreased, right? The tumor died. If you re-inject them, these poor mice, I mean, these poor mice, but if you re-inject them with more tumor cells, it stays down, right? These mice that were treated with the bacteria stay down, whereas the controls, I mean, obviously, if you inject them with more tumors, the tumor will grow, right? So it works in this model system. Now, I should say this is their best data. They have some other cell models, cancer model lines that aren't as good, and I'll show those too. So, for example, here, this is the breast cancer model, and it's not as good, as you can see, right? I mean, it's not flatlining, but it's still better than the one without the nanobody.
Yeah.
That's interesting. And again, this is injecting directly into the tumor.
And this is again with low, amounts, low stage. It's still not
Exactly.
a high stage.
Exactly. It's still starting off as a small tumor, and we're injecting it inside the tumor. The number of metastases, the number of cancer cell migration events also decreases, just like we saw before. One more cell line, melanoma. Are you familiar with what melanomas are called? Yeah. Skin cancer, exactly. So here's a skin cancer model, and again, you see it's again not as good as the original, but it is still significantly better than the other options. MAIA-3301 being the antibody against CD47 itself.
What was the first one again? What was the first model? Sorry, I forgot.
The first model was a B-cell lymphoma line.
Oh, okay. I see.
Yep. So it seems to work best for lymphomas. A little bit better, a little bit worse for breast cancer, and a little bit worse for melanomas. But it is still an improvement, I would say. Now, if you look here, we're going back to the lymphoma line, and this goes back to your point. We typically don't inject into the tumor itself, right? This is trying to just prove a concept that I can't actually kill the tumor. What if we inject it into the tail? Right here, they're sticking the mice in the tail.
Poor mice.
these poor mice. Oh my goodness. For science though. You inject it into the tail vein. So this is an intravenous injection, mimicking sort of the intravenous delivery of chemotherapeutics. And you can see how, again, even though it's not injecting into the tumor itself, the tumor does grow slower than the other groups. Which, I mean, what does that imply though, Angel?
That it's not as effective. Especially if you're looking, this is the same first graph that we saw,
Yes.
where it
Yeah.
fully flatlined and died.
Right.
Right. So there's a major difference, but it's still effective, like as you can see. But if it's not indirecting to the tumor, it's gonna grow.
Right. Exactly. So it seems like there is a injection delivery effect, right? But how about the fact that the tumor is growing slower at all compared to the other groups?
It just shows like it still works, even if it's not a direct. It's still effective at killing and inhibiting that "don't eat me" signal.
Right. Exactly. Which implies that the bacteria are localizing to the tumor cells, despite being injected intravenously and distributing all around the body. They still can target the cancer itself and cause it to slow its growth.
Yeah. The only thing I would think about is like a dose. Like, what is the dose being injected? Does increasing the dose, like, would it become more effective or would it just flatline?
Right.
Just some questions that I had over this graph.
Yeah, that's true. I don't have the dose on here, and I don't have it off the top of my head. But you're right. What you're talking about is the pharmacokinetics of this therapeutic, because the idea of pharmacokinetics is as we increase or decrease the dose, how does that affect delivery throughout the body? And also, as we increase or decrease the dose in the field of pharmacodynamics, is how does that affect, for example, tumor size? And so that would be definitely, if this does get into clinical trials, that would be definitely something they would have to look into. What is the optimal dose for different cancer cell lines?
Yeah.
Yep, definitely.
Another thing I wonder is like, okay, is the immune system building up a tolerance against the bacteria before the bacteria can localize as well? Is that like one of, this is just a theory of why it's not as effective even on the same cell line it was effective on previously. It's like as it's traveling, they pick up the whatever signals to attack the bacteria, and so they're killing it before it can like build, and then it's like hyper specific to the bacteria rather than letting the bacteria grow into the quorum that it needs to grow.
And that's really interesting. There definitely is an adaptive component to this. So the adaptive immune system is involved. I mean, I'm not showing the graphs. I didn't include graphs here, but they do show that the certain T cell effects are seen as well. So they're involved in here somehow. The thing about that is that these adaptive immune responses typically take a little bit longer.
Okay.
Does that make sense? And I'm not quite sure if, I guess this is a 20-day experiment.
Yeah.
So maybe. So you're right. So maybe there is a battle between this adjuvant vaccine effect and the immune system killing the bacteria itself. Right. Yeah. So that's a good point. Yeah. Good things to think about for sure. This one's really interesting also. So again, mouse, two tumors. What if we only inject one of them? Yeah. What if we only inject into one of the tumors? This is really interesting. Yeah. Okay. This is the tumor that they treated. They injected it into the tumor, and you see again, the one that had the nanobody dies.
I take back my next. I'll wait till the second graph. Nice.
The untreated tumor on the right side. By the way, this one that wasn't injected. It still shows a decrease in growth rate of the tumor, right? So that's
looks
super
similar
interesting.
to the intravenous model.
Yes, which also makes sense, right? Because if you think about it, the bacteria will travel to the other tumor by some passageway, maybe through veins, maybe through some other pathway.
Yeah.
But this is really interesting, and so this shows that this isn't just a localization where the bacteria are staying in one place. The bacteria travel, but they congregate in certain areas, right? And they congregate into the tumor. And this is just a graph showing the growth rate of the tumor. And so on the X and Y axis, it's just the growth rate of the tumors. And you can see that for the treated mice, the tumors are slowly growing in both the sticked and the unsticked tumor. So that's pretty interesting.
Do they ever show specifically bacteria isn't present in high quantities elsewhere?
They do.
Okay.
They do. In fact, I think that's the next slide actually.
Okay.
Yeah. Good. Really good point. Right here. The y-axis is a number of bacteria essentially. It's a measure of number of bacteria, and the x-axis is just location.
Yeah.
So they show that the treated tumor, obviously, a lot of bacteria, right? The untreated tumor has no bacteria.
That's interesting.
Right? So actually, I might have misspoken previously in which bacteria travel intravenously. It seems like they don't. Right? So in both the liver and the spleen, which are kind of controls in this case, there's no bacteria. In the untreated tumor, there's also no bacteria, which is really interesting.
I mean, you talk about like adaptive immune system. It's like, oh, once they start killing the cancer, do they pick up other signals that would allow them to kill cancer better?
Exactly.
Yeah.
Exactly.
That's cool.
So this is not just a localized event. This is a systemic, whole body anti-tumor response, which is super cool. And so the bacteria aren't there, but they're still causing the cancer growth to slow down. That's called an abscopal effect.
I think personally, that made this much better, in my opinion. Like, this is like, rather than just a localized, like, this is what the bacteria is doing, it's like you're also, on top of everything, you're modifying the system as well.
You're engineering the immune system.
Yeah,
exactly.
which is,
exactly.
it's like a side, like an adjunct. Would this be
adjuvant? Yeah. So it's
exactly.
like the adjuvant effect and also this abscopal effect, which is the idea that you have tumors somewhere and you have another tumor somewhere else, but you treat one and the other one goes away too. That's called the abscopal effect. So these engineered bacteria that undergo these quorum synchronized lysis cycles also have an abscopal effect.
Do the tumors have to be like the same kind?
That's a good question, actually. I don't think they try different tumor types, but if you think about metastasis, though, which is what this will be applied to in clinic, it will be the same type.
Yeah.
Right? That's a good point, though. If it would be different types, my guess is that it would be less effective.
Yeah, sure.
Yeah. Now, on these two graphs, what they're essentially showing is that the synchronized lysis circuit is needed for this decrease in tumor volume. So here they're showing just the circuit itself in green, just the nanobody producing bacteria in brown, and then both in red. And you can see that both the synchronized lysis and the nanobody are required for controlling this tumor growth in both the sticked and the unsticked tumor on the both sides of the mice. So what this says is that the synchronized quorum that allows bacteria to sense each other via that engineered circuit we saw in the very beginning, that's important for controlling this tumor growth. So all the parts are needed. And I love this quote. I mean, it's a bit jargony, but it says, taken together, all these results demonstrate that the engineered quorum lysis immunotherapeutic delivery system can generate potent, tumor-specific, adaptive immune responses that operate systemically to clear distant tumor lesions.
It's ASL.
I'm trying to give it some emphasis. And this is sort of their thinking as to how that works, the abscopal effect. So they're saying that maybe the bacteria induce not only the phagocytes that eat the tumor cells, but they also induce, as you mentioned, the adaptive immune system, and that's involved. They migrate from their lymph nodes and they travel to the site of the tumor to kill the tumor cells there. And so there's this immune system crosstalk that's happening between the bacteria, adjuvants, and the immune cells themselves. Super interesting stuff.
Yeah, I think that's cool. That's what I think.
Anyways, the main takeaway, the concluding statement.
I like the transformation graphic.
Yeah. The concluding statement is that you should not underestimate basic biology. I mean, all these things we talked about in terms of CD47 and quorum sensing, bacteria talking, if you think about that from like a basic science standpoint, you could ask, well, who cares about that, right? Bacteria talk to each other, so what? Right? Sure, there's a CD47, so what? But if you sort of synthesize and combine these ideas together, it can be really powerful. We can combine gene engineering, for example, like this cute bacteria eating this plasmid and turning green because it's the green fluorescent protein. I mean, these transformation principles and these gene circuit engineering principles can be used in very powerful ways to cause therapeutic benefit. And so hopefully that should be the main takeaway, is don't estimate basic science, and yeah, learn as much as you can. Any concluding questions or thoughts?
No, everything's good from there.
That's really cool.
That's really cool.
All right, we'll end it there. Thank you all for listening. Stay tuned for next week.
Later.